snap your fingers and stop like, do you enjoy watching sports? Snap your fingers and stop enjoying. You can't. 96 to 98% of the time.
Examining how, okay. Just got a message there. From the application of examining how cortical ensembles and networks actually determine which visual percepta may or may be perceived, and really try to gain an understanding of the network mechanisms that underlie perception. Of course, given that this is a neural interface seminar, I'm going to do a pretty deep dive into the technical side of the story. And so I'll get into those details. And of course, if questions are on that side or on the science side, I welcome questions on either side of the story. So to sort of frame the science and give some historical perspective on where these technologies, I believe, fit into the picture, you know, I just want to sort of put this in the context of, let's see if I can, there we go. You know, we've known for a long time, even from really early studies of neurosurgery patients that are receiving microstimulation of their cortex using electrodes. And remarkably, as many of you know, people will sometimes report having experiences in the case of perception. This is known as phosphenes. And in visual perception, this may take the form of flashes of light that people will say that they see, even shapes or textures, motion, and remarkably, even more specific and coherent experiences that may involve particular objects or even scenes. And it's really amazing when you think about this, given the way that this is actually done, an electrode lowered into the cortex, and we know from more recent work combining stimulation with calcium imaging, that really a somewhat arbitrary set of cells are being recruited by this approach. And so somehow the surrounding circuits and the brain is able to sort of interpret this information and ultimately give rise to particular percepts. Of course, individual percepts cannot be selected or reliably driven by these methods, but this tells us that there's this link between activity in these cells and perceptual experience. Now, this, of course, builds on you know, another element of the story which we've known for a long time, which is that activity in the cortex or in other brain regions shows very strong correlations with particular information. The classic example being orientation selectivity in visual cortex, that basically cells there will respond reliably to particular orientations of edges of light in the visual field, and this is true of a variety of different visual features. So what is the causal link between this? How can we actually gain a handle on this and directly select and which stimuli, which visual percepts will be experienced by the individual, and understand more about the underlying circuits? So more recent work, obviously still quite classical work from Bill Newsom and others, leveraged even using this somewhat coarse method of microstimulation, here applied to direction perception, direction of motion perception in monkeys, and leveraging the fact that there actually is functional organization anatomically of particular visual feature representations, they were able to show that they could causally manipulate behavior in the direction of the visual features that are encoded by these cortical neurons. And so this is, I think, really one of the very first and very nice causal quantifiable demonstrations that there is this link between these internal representations of visual information and the specific perception that in this case an animal is reporting. More recently, understanding how is it that activity starting in a particular cortical area could actually lead to what is likely to be a much broader network response that underlies perception. Using a similar approach here in a monkey, first training a monkey to respond to very weak visual stimuli and report the presence of these stimuli. These researchers first found, again, this perceptual threshold of these animals based on the contrast or the intensity of these visual stimuli, and then recorded across different visual and actually frontal areas simultaneously to show that when animals report seeing a very weak stimulus, activity appears to propagate across all of these regions. But interestingly, when the animal fails to see that same stimulus, when they miss that stimulus, activity is somewhat present in early visual areas, here primary visual cortex or V1, but that this activity appears to be very weak or die off in the propagation to other areas, particularly frontal areas. And so this suggests that there's this need for a more global network effect to be recruited to really give rise to perception. And in this study, they actually tried to give more evidence for this by, again, using this electrophysiological stimulation technique to stimulate the same visual regions. Here, finding the current stimulation threshold for when the animals would report seeing one of those phosphenes, which are those artificial percepts. And then again, recorded from, in this case, a higher visual area V4 while stimulating in V1, and found, interestingly, that it seemed to be required that activity would be propagated to a V4 in the case of very low current stimulation for the animal to actually be able to report seeing that stimulus. So again, this even more causally demonstrates that a more network level effect is required for activity that starts in V1, in this case, very directly with this microstimulation technique, to recruit that network and actually lead to a perceptual report of the animal. So even though this is, of course, very interesting and motivating work, it doesn't tell us really at a more detailed level how these circuits are organized that might actually give rise to these effects in natural vision. And so to sort of summarize what I've said so far, stimulation of arbitrary locations of visual cortex can cause artificial percepts known as phosphenes with specific qualities in humans. Activity in specific cortical populations correlates with specific visual features. This is obviously the Hubel and Wiesel work and many others.
That's a visual pointer.
Known for a long time. The stimulation of particular feature-specific populations, leveraging anatomical organization, can bias behavior towards that information. And again, that there appears to be this perceptual report depending on this network level recruitment. And this is known, at least in that paper, using the term ignition. And I'll kind of use ignition or network recruitment to sort of describe this type of an effect. So I think where the work I'll talk about today really fits into this story is in getting a handle on local circuit mechanisms that may select and amplify specific information to really select and amplify those particular perceptual experiences. And we can actually get, hopefully, a better handle on what those networks look like really at the level of single neurons within the brain. So this will move us into the mouse system where we're able to develop and use circuit level techniques at this scale. And there has been previous work looking at in reduced preparations in mouse V1 to map synaptic connectivity between cells that have similar visual tuning properties. And this is basically taught us that cells that have similar visual tuning, visual feature tuning, whether that be orientation selectivity or even other selectivities, are more likely to be connected with their neighbors that have the same tuning. And this has really been focused on layer 2-3 pretty much exclusively. These studies, which have shown this in a variety of ways, paired recording studies, EM tracing, rabies-based tracing, have all found similar sorts of effects and really point to the idea that perhaps there are sort of a cortical amplifier based on recurrent excitatory connections between cells that have these similar correlated responses, and that this may act to amplify that particular information and propagate it better throughout the broader network. But as I mentioned, this has really been focused on layer 2-3 in reduced preparations, and so we don't know, whether this is a broader feature that will apply beyond layer 2-3, for example, to other layers. And perhaps even more importantly, we really don't know if this or other circuit architectures are really related to this network ignition effect, this sort of nonlinear effect, this self-propagating signal that seems to be potentially very important for whether a perceptual experience is actually elicited and behavior occurs. So to gain access to these questions, I'm going to leverage all optical ensemble-specific interrogation. This builds on work previously combining calcium imaging and holographic optogenetics. I'll talk a lot more about that in a moment. which will allow us to select cells based on their functional properties, stimulate them very precisely, and then read out activity from those cells in the surrounding populations, as well as, as you'll see, monitor the behavior of the animal to see if we're having effects on the animal's behavior. So to do this, this involves combinations, as I said, of calcium imaging and optogenetics. And so we need to express both our calcium indicator, which is GCAM6, as well as our opsin here, carmine, as I'll talk about in a moment, a red-shifted opsin that we have found and characterized, which is here expressed in wide swaths of cortex very homogeneously by the expression from a single virus, which gives us this nice homogeneous expression, under a promoter that will direct this mostly to excitatory cells, and using a motif known as the KV2.1 motif that will actually target expression of that opsin preferentially to the cell bodies of these cells, and this will improve our resolution to really target individual neurons. And so you can see here the magenta is really localized largely around just those cell bodies. And you can see in vivo, again, that we have this nice, this is over a millimeter, we have this expression that is preferentially targeted to layer 2, 3, and layer 5 pyramidal cells, which are the output layers of the cortex. So we also developed a new microscopy system for stimulating very large populations of cells precisely in the mouse cortex. And this involves multiphoton imaging, which will give us access to that calcium, those calcium responses, which will allow us to characterize cells using somewhat, a standard resonance scanner 2-photon imaging. We've built on top of that custom optical elements that allow us to do this holographic stimulation. And what that really means is we can design the patterns of spots of light, as you'll see, that will be targeted to the specific cells that we want to stimulate. And that uses one of these spatial light modulator devices, which is a display device, a liquid crystal device, that will modulate the phase of the laser light such that we can perform essentially Fourier transforms to the lenses in the system to ultimately create the hologram or the interference patterns that will actually create those spots of light that are targeted to the cells of interest. This microscope system involves the integration of multiple spatial light modulators. And as you'll see, that allows us to overcome the limitations of any one individual device in terms of the temporal resolution of the ensembles that we'll be able to stimulate. And as you'll see at the end, we're also able to scale this in the spatial dimension to actually potentially increase the fields of view that we can address using multiple SLMs. So as I mentioned, I'll get into more of the sort of details of this, given that this is a neural interfaces seminar. So we actually worked with an SLM manufacturer to build a new SLM that's really designed in every possible way to achieve large addressable fields of view with rapid refresh rates and very minimal artifacts. And that involves the ability to temporally to control the temperature of the devices very precisely, a very high pixel count device with very high fill factor. So this really means that the great majority of our laser light will be diffracted by this device and will have minimal artifacts. This also allows it to handle very high laser powers, which is important for the number of cells that we'll be able to stimulate. And of course, there are a lot of electronics behind this to address all of these pixels very rapidly. And ultimately, pushing all of the boundaries on all these dimensions, this device can target, in theory, 1.6 by 1.6 millimeters field of view with the resolution of about a single neuron, which is about 0.5 NA, and has a refresh rate from hologram to hologram of about 500 hertz. And that means every couple milliseconds, we can actually change which cells we're stimulating just based on one single device. So to overcome that and actually achieve the kilohertz resolution that we want to be able to emulate any pattern of millisecond level action potentials, we integrated 2 of these SLMs into a single microscope and developed a series of custom breadboards and 3D printed elements, as well as custom optics to fold this into a compact device that can be mounted onto a commercial 2-photon microscope. and allow us to use these two SLMs either to stimulate more cells simultaneously or actually to interleave the stimulation to achieve that kilohertz overall ensemble update rate. One more piece to get the 3D recording of activity. We're actually using a piezoelectric device that moves the objective up and down very rapidly. So we can track the position of that piezo and use the spatial light modulator's 3D holographic capability to compensate for that real-time position of that objective, such that we can achieve our stimulation in a particular location in 3D, even though what that actually means is we're changing the location of that spot relative to the focal plane of that objective. So looking at what this looks like without the objective moving, this is what a stimulus will look like. Without that adaptation applied, the spot will move in and out of focus very rapidly. But when we apply this 3D adaptive control, we maintain that stimulation in the particular plane that we want to stimulate. So the other side of this is actually the redshifted opsin that we developed and characterized to provide a very robust and precise stimulation of specific neurons. So carmine, in comparison to other strong redshifted opsins, drives much stronger photocurrent and is much more light sensitive. So the amount of light that it responds to is far lower than these other opsins, meaning that it requires very low laser power, which is important for our ability to stimulate a lot of neurons. Under 2-photon stimulation, it is indeed redshifted. So now the wavelengths are essentially doubled. So 1080 nanometers appears to be around the peak response of carmine. And importantly, if we image essentially cells that are expressing carmine with the parameters that we use for GCamp imaging of 920 nanometer excitation, we don't see spiking. So we do see a bit of a modulation of the membrane, but we don't see spiking even at very high laser powers. So in addition to this very nice light sensitivity and strong photocurrent, carmine is capable of driving responses with very short pulses of light. And that again is important for the design that I mentioned. So pulses here with 1 photon light down at a millisecond or below are very reliable at driving active potentials in cells compared to other opsins. And indeed, when we stimulate cells with 2-photon light to really direct it to single neurons, we achieve about a millisecond of jitter in the timing of those driven action potentials. So this is basically exactly the type of precision that we want for a system like this. So putting all of those technical aspects together, this is an example experiment that we ran in mouse visual cortex, where we're characterizing this system. What you're looking at is layer 2-3 activity. It's pretty dramatically sped up in time. Those spots that you see flashing, those are individual neurons. And when you see these lines across the top of the movie, that's actually fluorescence. That's sort of an artifact that's created by our stimulation. So that tells you exactly when the stimulation is occurring. And when that happens, you'll see that there are constellations of cells that are simultaneously active at that time. And I should say this is over a millimeter squared of mouse visual cortex. So the way that we're doing this, we're actually activating dozens of neurons simultaneously with that SLM with very brief laser stimulation of 200 microseconds for each stimulus. And we're changing which cells we stimulate each millisecond to achieve this population control at kilohertz rates. And if I just show one example of this, so here, using that kilohertz capability, we're stimulating a couple dozen cells each millisecond, changing that each millisecond, as you see here in this sequence, repeating that at 30 hertz to drive a robust response in the cell that we can detect over time with the calcium imaging. And you can see that the great majority of these cells respond. And so this basically allows us to target populations of hundreds of cells, basically, or to write in particular sequences of activity that we may want to test. So, as I mentioned, this approach has 3D capability, So the SLM is capable of creating these patterns that can be directed in 3 dimensions to the particular neurons of interest. We've calibrated the system so that these spots of light that are generated that are essentially diffraction limited are really calibrated to the size of individual cells. This is a side view of cells from histology expressing the carmine opsin in their membranes, and then to sit to the same scale, a measurement of the point spread function of our system. So we can test this resolution in vivo by intentionally moving our stimulation off of our cells of interest. So this is an example of that, where we're taking a 15 micron spiral stimulation. So we take this spot of light and we very rapidly make a spiral around each of these cells simultaneously. If we move that spiral off of the center of the cells, we see that the response of the cells drops very quickly, and it essentially matches that diameter of that spiral. So that makes sense. We can do the same thing axially, so we can measure our ability to target cells in the axial dimension, which is a bit worse, as you can see, in terms of our optical precision. And in layer 2, 3, and also actually in layer 5, we're able to move that spiral intentionally off of the cell and see again this rapid drop off. Of course, it's not quite as tightly focused as in the transverse case, but again, still, for all intents and purposes, should really be targeting our identified cells of interest. So to look at how this all sort of pieces together, if we now display the calcium activity, this is actually changes in calcium activity that you see playing in this movie, of cells that we're directly targeting with the holographic approach. Here are circled with these green circles. And you can see that we drive these robust responses in those cells. Very interestingly, we also see that there are a few other cells in the surrounding population that also respond. And as you may imagine, we were very concerned, are we accidentally striking some of those cells? Of course, there are cells expressing opsin throughout the volume. We're doing all we can with targeting opsin expression to individual neurons and calibrating our optics. But as you'll see, we do a series of analyses that really ask questions about specifically those cells to find out if they really are just sort of arbitrarily, hit cells, stimulated cells, or are actually showing us evidence of functional connectivity that we're actually recruiting in that surrounding population. So this is actually across cortical layers. This is across layer 2, 3, and layer 5. And importantly, the way that I've actually selected these cells that are circled in green was based on the fact that those cells were strongly correlated with a particular visual stimulus. And this is the sort of basic concept for a lot of the data that I'll show next. So basically, we want to ask if we stimulate cells within V1 that have a particular visual tuning property, are they going to potentially recruit other cells that have a similar tuning or similar dynamics that may actually underlie some of those ignition-like effects that I talked about in the introduction. So to gain some ideas of that, we imaged cells across the cortex, as I just showed, and found cells that respond specifically or preferentially to particular visual stimuli. So here, cells that respond to these vertical drifting gratings, but don't respond to orthogonal gratings. These are sort of traditional orientation-selective cells. You see they have these selective responses. They respond to one, but not the other visual stimulus. And to use a control to compare against for our analyses, we also selected or chose sets of cells that have the same number of cells as these tuned ensembles, but are selected at random from the surrounding population. And again, these are cells that are not necessarily tuned, but will act as a control for our analyses. And so we can stimulate these cells directly and drive robust responses in these neurons directly. And then to see if we really are triggering dynamics in the surrounding population that resemble visual-like dynamics, we're going to do a series of analyses on the unstimulated cells. So we're going to exclude all of the stimulated neurons. And just to be safe, we're going to exclude cells that are neighboring those cells, as well as any cells that are directly above or below those cells. And that'll give us a population to look at, to cleanly look at the functional recruitment of cells that are not directly stimulated in any way. So we do a series of analyses that look at the surrounding dynamics to try to compare visual-like dynamics that are visual dynamics that are basically evoked by visual stimuli, and then see if the tuned ensemble or random ensemble stimulation cases resemble those visual-like dynamics in any way. And so in this case, we use a logistic regression analysis to develop a classifier that will classify the visual conditions based on visual-only trials. So there's no optogenetic simulation on these trials, to basically determine these logistic regression weights and use it as a classifier. If we look at cells that have high weights in this analysis, they indeed are cells that are essentially tuned for one of the other stimulus. So they respond preferentially to one 0 degree or 90 degree stimulus. Those are those different oriented gratings. And if you look at the difference of that, we see that these populations really do essentially discriminate between these two visual stimuli. Now, if we take this classifier and we don't change those weights in any way, we do no additional training, and then apply that same analysis to the other conditions, now the held out visual cases, tuned ensemble stimulation, random ensemble stimulation, or no stimulation of any kind, we can then look at how our classifier does and how these cells respond. So if we look at these cells that had high weights in this analysis, we see that during tuned ensemble stimulation without a visual stimulus, that again, we see similar responses, or at least many of these cells show strong responses. And again, there appears to be a preference in this, when we look at the difference in their responses between these stimuli, where the same cells that responded during visual stimulation appear to be recruited during tuned ensemble stimulation. And we don't see this during random or no stimulation. So if we look at the performance of our classifier, so actually using those data to classify the different trial types, with held out visual only trials, the classifier does a very good job of classifying those conditions, and can also classify, based on this unstimulated activity, the unstimulated responses in that surrounding population. It can classify which tuned ensemble is being stimulated, either the 0 degree tuned ensembles or the 90 degree tuned ensembles. But this is not true for the random stimulation cases. So this tells us that there is actually discriminating information that actually is along essentially these dimensions that are defined based on that visual only data. And to get a more qualitative picture of that or understanding of that, we performed a principal components analysis, which has a similar sort of concept, where we define our principal components based on the visual only trials, which as expected shows these trajectories over time. This is over the time of each of these stimuli that separate the 0 degree and 90 degree visual stimulus conditions. And when we look at the other ensemble stimulation conditions, while not changing those principal components, we take those visual principal components and project the unstimulated neuron activity onto those dimensions during ensemble stimulation. We see that during tuned ensemble stimulation, but not random ensemble stimulation, that we have a qualitatively similar a pattern or a set of dynamics that are recruited in that surrounding population. So taken together, these data basically indicate that when we stimulate these functionally defined populations that are defined based on their visual properties, that they themselves are capable of actually recruiting local patterns of activity, network responses that seem to amplify this particular visual feature. And of course, that leads to the question of whether that is sufficient to actually potentially give rise to a particular perceptual experience for the animal. And so to really ask that question directly, we then put these mice, which in this case, as you see, we call them naive mice. They haven't been really trained in any way. We moved mice like these into a visual discrimination task, so we can actually ask the mice, to the best extent that we can, what they're actually seeing. So in this case, first the mice are trained to discriminate between these same visual gratings. So here the animals learn to respond with a lick to a water spout to get a water reward. They're water restricted. So they get this reward if they lick during vertical gratings. And they learn to withhold licking on the no-go trials, which are horizontal gratings, to avoid a penalty or timeout or an air puff if they try to lick the port during that stimulus. The animals learn to do this very well, so they achieve a very high D prime, or upwards of 90% correct after several sessions over a variety of visual contrasts. And we find the point that the animals are no longer able to do this based on the visual stimulus, basically the contrast or the intensity of that stimulus that's just below that perceptual threshold. And so we did this to sort of first try to replicate the experiments, the classic Bill Newsom experiments, where to see if we could actually modulate the animal's ability to detect and discriminate very weak visual inputs to the animal. And we figured this would be the most likely thing to work, given that we're simulating a relatively small fraction of the overall population. And so if we find cells based on their visual tuning properties as we did before, define random cells as we did before, and then stimulate each of these ensembles in combination with these different visual stimuli or no visual stimuli at all, or no optogenetic stimuli at all, we can compare the visual perceptual threshold of the animal, which is here shown in black, where this is the visual only trials where around 3% contrast, the animals are able to begin to do the task reliably. Specifically, when we stimulate the tuned ensembles, the animals increase their ability to do this task. They're able to do this basically at lower contrast. And there isn't a consistent pattern that we see during random ensemble stimulation. So this basically fits with that same concept of being able to modulate behavior by targeting specific visual feature information in visual cortex. But what about actually driving potential visual percept without a visual stimulus present? So can we actually altogether drive a visual experience for the animal directly from these ensembles? And so for this, we actually turned off the visual stimulus altogether, and again stimulate these particular tuned ensembles or random ensembles, and observed how the animal behaved. In this case, the animals actually were able to transfer that visual discrimination behavior to the ensemble discrimination behavior. So if we look at the performance of the animals actually after that experiment that we just ran, which is actually mapping that modulation of their visual contrast perceptual threshold, the animals were able to very robustly actually do this task. Each of these X's is a different mouse. And they were able to do this across time. So this was over the course of at least 2 days, and I'll show over a much longer time frame in a few slides. So this is with the tuned ensemble stimulation. We of course have our other set of stimulation, which is the random ensemble stimulation cases, which are randomly interleaved trials in these sessions. And in this case, the great majority of mice could not perform the discrimination with random ensemble stimulation. But very interestingly, after some time of doing this experiment, eventually a couple of animals actually were able to do the discrimination with the random ensemble stimulation. So remember, these random ensembles were not tuned to the visual stimulus when we initially recorded from these volumes. And yet, apparently, through the combination of perhaps pairing with the visual stimulus and some incidences of reward and punishment, the animal has, some of these animals anyway, have been able to learn to actually use that random ensemble activity to perform a similar task. And so to get a better handle on, okay, well, really how special, are these tuned ensembles, these tuned subnetworks? We wanted another control to sort of, give us a greater understanding of this. And so we moved to an entirely different field of view, a different set of neurons, by moving axially in the volume, and defined totally new sets of tuned ensembles as well as random ensembles that were never previously stimulated or paired with reward or anything like that. And then we put the animals immediately back into the task. And when we do this, we see that the animals are able to do the task to high performance with the tuned ensemble stimulation, but not the random ensemble stimulation, these new ensembles. And so the animals are able to generalize the discrimination of tuned ensembles, but not random ensembles. Basically, this tells us that we can move around that tuned population. You know, it's not just those specific neurons that we had initially targeted, but actually a more general concept of hitting these particular functional classes of cells. And clearly, apparently, and as you'll see some data, through some plasticity or other effects, the animal is able to, in some cases, the animals are able to take random ensembles and actually link those cells to these behavioral networks. So as I mentioned, you know, I showed examples of a couple sessions there. We were able to actually target these ensembles and populations over many sessions. So this is showing, you know, from one of those volumes, an optical slice in layer 5, where we're returning to these same exact populations day after day for many sessions. And zooming in on one region in layer 5 and overlaying the first session with each subsequent session, we can see that we maintain this single cell resolution across the course of a month of imaging. And actually this spans even 2 months of actual calendar time. We also implement during sessions online motion correction. So we're actually reading out those images in real time, comparing that with cross correlation to our starting images, our target images, and then moving, essentially, you know, moving a bit the stage and the animal to maintain that alignment over the course of the session, so that we target those cells that we want. And this shows, you know, over the course of these sessions, actually the data over 2.5 million images, 9 mice, and all of the sessions that from all the data that I'll show today, where we maintain for greater than 90 or 95% of all of these images within 5 to 8 microns of the original alignment. And so that gives us confidence that we really are targeting those specific cells. And of course, that's critical for being able to drive these reliable behavioral effects. Indeed, if we look at the behavior over the course of a month, returning to those same ensembles, here shown in red, showing the animal's performance in discriminating these tuned ensembles, you can see that their performance is approximately 90%, which is the same high performance that they achieve with randomly interleaved visual stimuli. So there's basically no difference between their behavior on visual or ensemble discrimination trials, and they're essentially saturating at about 90% correct. So we can take all of these data, which generates a lot of data, you know, over 100 sessions, to again look at what the neural dynamics are in those unstimulated populations, to see again, you know, what really, you know, what type of dynamics are we seeing that may resemble visual-like dynamics during that tuned ensemble stimulation case. So here again, applying our classifier analysis, decoding analysis, comparing the behavior of the animals to the performance of our classifier, we see that on held out visual-only trials, that there's very high performance of the animal as well as our classifier. And this is true of discriminating or decoding tuned ensemble stimulation cases. Very interestingly now in this behavioral cohort, we can also, to some extent, actually decode the random ensemble stimulation cases, particularly for animals that have learned a bit to do this discrimination based on those random ensemble stimulations. So now versus those naive cohorts where there was absolutely no decoding performance, here there's still this there's this essentially one-to-one relationship between the classifier's performance and the behavior of these animals. Looking at these PCA analyses, which show us neural trajectories, actually each of these traces is an individual session. So you can see that there's many sessions, as I mentioned, basically over 100 sessions that we're showing data from here, where these principal components are based on visual-only trials and show the diverging dynamics that occur during visual stimulation. That a similar divergence of these dynamics is seen during tuned ensemble stimulation. And now, interestingly, whereas most mice show no modulation during random ensemble stimulation, we see that for animal, in this case, one of these animals that had learned this discrimination, that we see a bit of a mapping of that activity during random ensemble stimulation onto these visually defined components. And so that again, that sort of indicates that there's this specific plasticity that appears to have potentially linked these random cells to these visual-like networks. So we also looked at sort of a functional connectivity level analysis, which I think underlies all of these effects, basically demonstrating that if we look at the tuning of those other cells in the surrounding population that are recruited during ensemble stimulation, that they're far more likely to be similarly tuned with those tuned ensembles than orthogonally tuned. And so basically, if we map the tuning of those recruited cells, we see that it's over double the probability that those cells are isotuned or of the same tuning as the stimulated populations. So we used this analysis to actually try to add on to that earlier work that I mentioned in the introduction in terms of the populations that are recruited within and across cortical layers. So to do this, we basically subselected or selected a subset of those tuned ensembles to stimulate on individual trials. So here, for example, we stimulated only layer 2-3 neurons while recording from layer 2-3 and layer 5, and then looked at what cells were recruited across those populations. And indeed, when we stimulate layer 2-3 tuned ensembles, we see that there is this same pattern that has been observed by other methods within layer 2-3, that there's a preferential recruitment of similarly tuned cells within layer 2-3. And we add to this story by showing that actually this also is true of the recruitment across layers to layer 5. We see recruitment of layer 5 activity, and it also shows this preference for cells that are similarly tuned. If we do the same experiment, but now actually stimulating only layer 5 neurons, we find that, again, we find this feature that similarly tuned cells are recruited more so than orthogonally tuned cells. But interestingly, we don't see much recruitment of layer 2-3 activity, and there doesn't appear to be any preference in the recruitment that we can detect from this particular functional connection. And so this sort of demonstrates, again, this sort of repeating pattern of this potential amplification type of a circuit that has this sort of directionality from layer 2-3 to layer 5 that sort of matches with the canonical circuit of cortex. And to get an even greater understanding of how this sort of preferential recruitment of layer 5 may have relationship to behavior, we actually looked at the difference in these trials with the animal's ability to actually do the discrimination task based only on stimulation of those cells in each of those layers. So here we're actually intentionally reducing the number of neurons that we stimulate, stimulating only cells in layer 2-3 or layer 5. And we find that at around 20 neurons, the performance basically saturates. The animals perform quite well. They can do this with stimulation of either layer, but then it very rapidly begins to drop off as we reduce the number of neurons that we stimulate. And this happens faster for layer 2-3 than layer 5. Another way of saying that is that we can stimulate fewer neurons in layer 5 to drive behavior, or that cells in layer 5 appear to be more potent at driving behavior than layer 2-3. Interestingly, if we look at our classifier analysis, our decoding analysis, again of the unstimulated population, there's a very similar pattern. Basically, at approximately the same number of cells and with a similar sort of relationship, the decoding performance drops off very rapidly. And this correspondence basically indicates that once that discriminating information is detectable in that surrounding network, the animal apparently is able to use that information to do the task. And again, underlying, I think, these effects is this selective recruitment. So here showing the proportion of isotuned cells in that unstimulated population that are recruited depending on the number of neurons that we stimulate. And again, we see this nonlinearity at around the same number of cells, where there's a very rapid sort of increase in this recruitment that appears again to correspond to the behavior of the animal. So we've, I believe we've added, information about how selective recruitment of percept-specific populations and dynamics in V1 it actually exists within V1 directly from neurons within V1. This favors propagation between layer 2-3 and layer 5, and layer 5 is more readily driving behavior. It's more potent. So we've added more information about what this underlying circuit actually looks like and fundamental features of it. There's this tight relationship, as I mentioned, between what we call critical dynamics, or this nonlinearity that we see in sort of driving these widespread network level effects that correspond so tightly to behavior, which if you think about it, really amazingly seems to point to a more brain-wide network that must be recruited for the animal to actually do the behavior. So this fits with the prior work that I mentioned in the introduction, where it appears that there needs to be this this ignition or this broad recruitment of a broader network to actually elicit a perceptual experience and drive behavior. So in terms of so that's really the science, and I think it shows that using these types of approaches, we can actually, now actually gain network level access to questions and hypotheses about how circuits are organized and actually implementing transformations that are relevant for perception and behavior. We've of course also developed and demonstrated this all optical neural interface, which has interesting properties. I showed that we can basically stimulate dozens of neurons per millisecond in kilohertz sequences, volumetric addressability across mouse cortical layers, and we can read out activity from thousands of local neurons relatively slowly, on the order of a few hertz. It's actually about 3 hertz or so. So certainly much slower than our capabilities with stimulation, sufficient for reading out those general broad calcium dynamics, but certainly nothing close to what we would really want in terms of millisecond level resolution. But this is importantly, apparently sufficient for driving robust network and behavioral effects. And so we hope that this will be useful for many people in studying many different systems. So, where might this go in the future? So, how scalable are these approaches, and really how could we potentially improve the optical activity readout, for example, to achieve that temporal resolution? So in terms of the spatial scale, I mentioned that we're targeting a millimeter squared with this approach, and that the macro SLM has a 1.6 millimeter theoretical addressable field of view. That would do a bit better, and you can sort of get a handle on these are different visual areas drawn out on mouse visual cortex. You can see that we're meaningfully targeting regions within the cortex. Of course, we'd want to go potentially even bigger than that. And it's worthwhile mentioning that we actually have demonstrated the ability to multiplex not only in time, but here actually in space, using an earlier version of the SLM, a much smaller field of view that was originally targeted by these earlier devices, where we're tiling in space essentially their addressable fields of view using a tilting prism that's inserted into the optical path. And so this in theory can allow us to maybe even quadruple, or obviously in practice maybe somewhat less than that, but actually increase the fields of view that we can target. Obviously, the optical setups become more complex, but there is this potential and in principle possibility to expand these technologies to address larger fields of view without needing to necessarily develop bigger and more complex SLMs themselves. In other work, not using SLMs, but sequential stimulation, we've demonstrated that we can read out and stimulate single neuron activity through a grin lens, which gives us access to deep brain structures. For example, orbital frontal cortex, shown here, where we're able to modulate feeding behavior by stimulating feeding responsive cells. And in principle, SLMs could be used here to actually apply this in 3 dimensions and stimulate more cells in more temporally precise ways. In terms of improving the imaging side of these technologies, there's been a tremendous amount of work in the field. This is showing, this is sort of a summary of many different labs work to expand by multiplexing or other methods the ability to image across large swaths of mouse cortex or across layers, including work from Mark Schnitzer's lab here. And his lab and others have also pushed the multiphoton imaging capabilities even into the kilohertz regime. So for the most part, these are often no faster than tens of hertz imaging, but it is possible actually to do two-photon imaging with kilohertz resolution. Also, a very inspiring and important work from Michael Lin's lab here at Stanford. So, developing and applying voltage sensitive indicators that are multiphoton compatible. So here in a very recent study that I think is very important for the field, showing that it's possible to actually record individual action potentials and even subthreshold dynamics in mouse cortex. Others have applied this even in mouse visual cortex to map out things like visual tuning based on spiking and subthreshold activity. So this type of approach could be imagined for actually getting some of that millisecond level resolution that we really want. And even early on with these all optical methods, it was realized that the ability to stimulate individual neurons and record from other cells, even with an electrode, can take what is normally a multi-electrode experiment here, a paired recording study to map particular functional synaptic connections between cells, that can be made a more optical and higher throughput experiment. And perhaps even with voltage indicators, we can imagine someday being able to remove all these electrodes altogether to really increase the throughput of these approaches. So in general, I think it's exciting to think about the future potential for readout at millisecond timescales, for more direct emulation of increasingly realistic activity patterns to understand how they relate to particular experiences and behavior, and to actually gain more direct access to connectivity. The connectivity I talked about today is a functional connectivity, a slower, a resolution look at what is recruited, but of course, ultimately we would want a much more precise measure of that. And so with that, I just want to thank many people who are obviously involved in this work, in particular Yoon Kim, Tim Machado, and Sean Kieran, as well as many others in Carl's lab, as well as Surya Ganguly's lab, and others who have contributed to making this possible, and the various funding sources. And I would be happy to take any questions.
Thank you very much, Jim. Very interesting, impressive optics, and very interesting neuroscience. So really enjoyed your talk. So let me just remind everybody that we'll now unmute or allow everyone to unmute themselves when they want to. You don't have to if you don't have a question. And please type your questions or short versions in the chat, and then either myself or Jim will be able to unmute you. And also I suggest you turn on the camera when you ask the question. It will make it more personal. So while you are typing, maybe let me ask just the first quick question. What do you think is a perception? These mice, they don't see an image when you introduce this sort of 20-cell stimulation. I wonder, and yet they make a perceptual or behavioral decision. What do you think is perceptually visible to them?
Yeah, well, we don't know exactly, obviously, but the readouts that we have are, one, the behavior of the animal, which we see this transfer, very robust behavior that resembles their visual behavior. It's generalizable to the different populations that we stimulate. And then we have the neural dynamics. So which is really a new view, I think, that that hasn't really been possible before. We can actually see that it isn't just any activity that's sort of recruited. It's really resembling the activity that is evoked by visual stimuli. So that gives us access to the internal representation of visual stimuli, of visual perception, that gives us more evidence that what they're experiencing has a sufficient similarity to those visual stimuli. We're expanding, of course, on this and trying to come up with ways of gaining even more, even a greater understanding of what they're experiencing. For example, like flipping the experiment where we train them to particular ensemble stimulation and then look at their behavior to visual stimuli. So can they transfer in the other direction? And very early data suggests that is possible. We've at least seen one example of where this is true, where an animal can transfer from a particular ensemble stimulation to visual discrimination. But it's one of those things that in animals, it's going to be very difficult to know for sure exactly what they're experiencing. And we really rely on their behavioral readout, and now actually these neurodynamical readouts to gain as much of an understanding as we can. And I think through further development of each of those domains, also the readout as well as these behavioral readouts that we can gain more understanding of what they're experiencing.
Okay, very cool. So I see there is a question by Charles. Let's see. Karina, can you unmute Charles? There's a long list of guests here and I don't see everyone.
Yeah, thank you very much for the wonderful talk. I'm just having a quick question regarding the future direction of this technology. So can you optically and selectively inhibit neurons instead of stimulating them?
Yeah, that's a great question. We're very interested in that, and others, of course, in the field have been as well, and have shown, there has been evidence that is possible. We didn't focus on it here. We're kind of working on that now. There's a lot of, typically, we were quite lucky to find this carmine opsin, which is very sensitive and robust, as I mentioned. Inhibition tends to be a bit harder. We need to inhibit the cells for longer periods of time, and the wavelengths, have to be worked out and all of that. But in principle, yes, it is possible. And I think it would really add to our ability to ask, how necessary these particular activity patterns are for perception.
Well, I don't see other questions yet in the chat, so maybe I'll ask you another technical question. So the computation of holograms is actually quite laborious and takes time. And so you mentioned something like real-time correction for tissue movement and so on. I'm curious, you must have very powerful computer to be able to do it in real time, based on my experience, or these are really shift corrections which are easy to compute.
Yeah, so in that particular case, that is relatively simple calculation, but it still does take time. The whole loop is on the order of tens of milliseconds. But in this case, because we're imaging at that slower rate and we're actually imaging a volume, that is sufficient. And what we're really correcting for there are more of these slower drifts over the course of a session. The more rapid changes, or shifts that happen when they're running, for example, they're much smaller, thankfully, because we sort of address that more with the stability of the overall preparation. So they're on the order of a couple microns, which is sort of, we sort of live with that kind of wiggle in our preparation. Of course, that's corrected offline for all of our analyses very precisely, but online currently, we're currently in that sort of tens of milliseconds closed loop. We're working on going faster than that. So we have access to the images on the order of 5 milliseconds or so. And the original idea for those SLMs, and I sort of quickly mentioned the underlying electronics, was that we would have a closed loop capability to actually change or decide upon what ensemble to stimulate within milliseconds or very few milliseconds. And that is ongoing work to actually have that kind of throughput to the device itself. And yeah, indeed, it does require, everything, all computers involved and everything involved being, running at this very high rate. But we haven't fully leveraged that yet.
Okay, so I see there are other questions. So I suggest whoever is asking the question, you can unmute yourself, but also maybe activate the camera so it will be more personal. I myself have to sign off, so I apologize, but Jim and Karina, please carry on and manage the meeting. Thank you very much again. Excellent.
Thank you.
Thank you.
Karina, will you be unmuting people, or shall I? I don't see that.
Yes, I was unmuted. I have a question about this few mice that were able to learn from the random stimulation. Mm-hmm. Do you notice anything generalizing age-wise, gender-wise, anything else that you will be able to predict what mice will be in this small group and what mice will not?
Yeah. They were all males in the study and of approximately the same age. It is a long-term study that actually, what you saw today was several weeks worth of data, essentially. So we don't really, we can't really say anything regarding that. But, we don't know for sure, but perhaps there's something about, we select the cells at random, but perhaps those cells, by chance had, some underlying properties, some existing functional properties that made them more likely, some connectivity that existed already, for example, that made them more likely to be integrated in this way. Of course, this is all anecdotal, but in general, not all mice are the same, even when they're learning the visual task that, they learn at different rates, or, perhaps there's just, something about their ability to sort of interpret this information. I think that, it was one of these results that we were able to really demonstrate, significantly for individual mice, but I think is going to take quite a bit more work to really understand, when that happens, what are those parameters that may more preferentially, drive particular plasticity, for example, what are those temporal windows, what patterns of activity or what combinations with reward or other things are necessary to really not only create plasticity, but you have to, think that it has to be something that that sustains over time, so that the animals can actually use this in a behavior. And so I think it's in some ways a whole different direction to go in terms of understanding, circuit basis of learning and things like that. But in terms of what was different about those mice, I think, we don't know actually. I think we'd have to perhaps do more analyses or even a full-scale study to really ask that question.
But you did not proceed with them to different behavioral tasks to see if they will learn faster, better, independently of the task.
Yeah, so we did the, one experiment, which was importantly to go to new random ensembles and show that they couldn't transfer that behavior. I mean, maybe eventually they would be able to at some point, but they weren't able to do that in the time that they clearly demonstrated the visual discrimination. I think in sort of implicit in your question there is that one that I find really interesting, and I hope to be able to follow up on someday, which is, if again, other dimension, which is, understanding properties of plasticity in circuits and cortical circuits, can we actually manipulate the time it takes for animals to learn a particular task? In a way, we may have tools here that give us access to these types of questions. Is it actually strengthening of connections in these particular, in any particular network that actually are underlying a certain type of learning? I mean, that's just a general question that is hard to ask. Here, perhaps we show some evidence that there is plasticity that appears, goes along the dimensions you may have expected, and now, sort of post hoc looking at the data. But whether it allows them to transfer to other behaviors, again, I think that sort of requires a whole new study.
Thank you.
Hi, James. Thank you for the very interesting talk and the cool results. I just have a question about the design of experiments. If I understand correctly, when you train the mice, you pair the opto and the visual stimuli together. So for this design, my question is, how do you rule out the possibility that when you train the mice actually learn to associate this stimuli with the visual stimuli instead of it's actually these neurons that's coding the visual stimuli?
Yeah. So again, to kind of review, so the first data that I showed without behavior and without any pairing of visual, actually had no pairing of visual stimuli and optogenetics, or as I mentioned, reward or punishment. In those experiments, we also saw, at least for those dynamics, those trigger dynamics, that we saw those visual-like dynamics in those surrounding populations. So that appears to be something that's even present in naive animals where there isn't really any behavior. The animals were awake, they were sort of running freely on a ball, viewing these stimuli passively, but they had no understanding of that those stimuli meant anything to them. So that's one element that certainly gives us very strong evidence that nothing about our stimulation is actually directly causing that particular pattern. In terms of the behavior, as I mentioned, there's even in the original sort of experiment, we saw a much more robust and reliable effect in the tuned ensemble stimulation cases than the random ensemble stimulation cases. It was indeed true that one or two of the mice were able to actually eventually do the random ensemble stimulation discrimination, but that was much more apparent and much more immediately obvious in the tuned ensemble stimulation cases. Also, I described that we actually did a very important control experiment where we moved to entirely new sets of tuned ensembles and defined new random ensembles. And so that, after the animal, presumably there's some difference, obviously, that the animals need to understand that, they're not exactly seeing the visual stimulus, but to behave in the same way. And so we sort of think that after they've sort of, done that behavior, that they're able to do this tuned ensemble discrimination very nicely and reliably, that then when we go to these new ensembles, they're able to generalize that very rapidly. That basically, in the course of that first session, we see that the animals are able to do that discrimination, whereas with the new random ensembles, they can't. And so that shows us that with new random ensembles, even cells that have never been paired with visual stimuli, that they're able to do this generalization. And so that gives us, I think, the strongest evidence that there's truly something special about those tuned ensemble stimulation cases that doesn't require this pairing or that particular type of possible plasticity that may occur in order for the animals to generalize visual discrimination to the specific visual tuned ensemble discrimination.
I see. Thank you.
Sorry about that. You talked a bit about models in your introduction, and you mentioned recurrent connections. I'm curious to hear your hypothesis of the neural mechanism, what it might look like, how maybe inhibitory cells, to the point of, where do inhibitory cells come in, and how do you think the whole thing is working? But it's a huge question, but just kind of your intuition.
Yeah, so here we really focused on excitation. We had this hypothesis going into it based on that prior connectivity mapping work from others that there may be these, there certainly are recurrent excitatory connections within layer 2-3, and that might act as sort of an amplifier within the visual cortex, and so that kind of formed the basis of our hypotheses for this study. Do we see evidence of that in vivo? Does that have any relevance for perception of behavior? And so I think we have evidence of that here. What is it useful for? We focused on kind of a weak stimuli. This is sort of at the perceptual threshold of animals. We saw that we could modulate that, and that that's been known from the classic work I mentioned. So perhaps if there's in a noisy environment or where there's a lot of information coming into the system, this can help actually filter that information and propagate cleaner signals going forward. And in terms of the critical dynamics that I described, which is really this nonlinear effect that we find that we stimulate just enough neurons, and then we sort of see this widespread network recruitment occur in a very sort of nonlinear way. That gets to that more ignition-like effect that I was referencing in that earlier work, that suggests that just sort of an all or none, or sort of this self-propagating signal that takes that activity that otherwise might be thought of as in sort of a vacuum in early visual cortex, doesn't really lead to any sort of network level effects. But then when it recruits that even those local cells that we can see, that at that same time or very nearly that same time or amount of cells, that also is apparently recruiting a much broader network that must be necessary for the behavior. So that I think brings up a lot of questions about really what is that more brain-wide network? How does that happen? How is it that you can start from 20 neurons and have it really recruit such a broad network? Does that are there sort of repeating amplification motifs that are repeating across the cortex or other areas, that resembles what we see in V1? Those are just like the types of follow-on questions that I think, we and others may try to ask. In terms of the inhibition, that of course is a very interesting set of questions, and we're actually working on that now. So we're labeling interneurons at the same time. So we're using a slightly different virus that allows us to express in excitatory and inhibitory cells, while a particular class of inhibitory cells is labeled with a red reporter. And that hopefully will allow us to read out and stimulate different patterns of excitatory and inhibitory cells. And the idea is, perhaps we can look at whether particular interneurons, have, roles in gain modulation or gating, for example, that has been implied by earlier studies, that might actually help determine which activity in, say, V1 is gated or propagated or some modulated to actually propagate to other areas. And so that's one direction we're going in terms of inhibition, is trying to see if we can sort of a hypothesis of, based on those different types of cells, that they may be involved in gain or attention-like effects that influence that threshold that we see. So hopefully, yeah, that addresses your question.
Thank you.
Hi there. Let me see if I can start my video. So my question is a little bit more on the technical end of things. How do you generate the appropriate spatial patterns for this sort of holographic stimulation a priori without knowing much potentially about what stimulation patterns would be relevant? And what also happens if you have different efficiencies of opsin integration within that area of tissue, you may not get the spatial pattern that you wish. Yeah, so the way it works, and I couldn't really get into the details, I guess, but, basically the spatial light modulator creates this phase mask that modulates the phase of the laser. The laser is actually coherent lasers or light source. So all the incoming light is of the same phase, and so we're in a spatial way modulating that phase. We sort of do an inverse Fourier transform of the target pattern to display on that SLM. When you go through a lens in these optical systems, you're actually essentially performing Fourier transforms so that we can actually we can very nicely predict what pattern we're going to generate in the sample. So we're sort of going backwards through the microscope, and so we can generate those patterns in that way. That's basics of how it works. In terms of the opsin expression, that's a great point. So that was one of the big steps. We did this a lot previously with expressing the opsin and the calcium indicator via separate viruses. We would then mix them together and kind of create this cocktail. And we would see, we would see, kind of overlapping patterns, but there might be some even competition, we wondered sometimes, and things like that. And so it really was having both the calcium indicator and the opsin in the same virus that helped a lot. So that there's this P2A element between those two genes that has been shown by others, and actually we confirmed in our own data, leads to expression of both, 100% of the time, basically. So in all the hundreds of cells that we counted in histology, they had GCAMP, they also had the opsin. And so that we think has really leveled the expression levels across the populations of cells. So we think that we have, relatively homogeneous patterns. I mean, the other readout that we have is just how many cells that we stimulate show robust responses. And in general, we see something like, almost 80% or so give us a robust and reliable response. And so, perhaps there's a little room there to get it to 100%, but for the most part, I think we have sufficient expression to be driving spiking in the great majority of cells. Okay, well, if there are no other questions, again, I really appreciate everyone who joined via Zoom today and the opportunity to give this seminar today. Thank you very much.